Okay, so we are recording. I have to try to get through a ton of stuff in this meeting today, so I will be probably moving and talking more quickly than normal. I can't review, dissect, and talk about every single little tiny point in all these sections, so I have to just sort of plow through and do my best to get through what I think are the best, simplest points to allow you to understand the detail on top of that. So that's my goal. So without further ado, I'm going to call up my first whiteboard and just jump in where we left off yesterday. So I will do that. Yesterday we talked about resting membrane potential. That's where we left off. When a nerve cell is at rest, what is the environment of the cell? So I'm going to draw a cell. What I'm leading up to now, the first main topic today is how that neuron generates and transmits its own electrical impulse called an action potential. So that's what we're gearing up for today. So I'm going to quickly draw my neuron, my motor neuron cell. I don't have time to repeat the whole resting membrane potential business that we left off with yesterday. Make sure you either listen to or watch that lecture first before this one, because that information is necessary to really make sense out of what we're going to talk about next. So here's it's actually a typical motor neuron. I'll put a small nucleus in there just to remind you that that's there. So we know that sodium is in high concentration outside of the cell, potassium is in high concentration inside of cell, and that goes all the way around this membrane, this plasma membrane. I'm just not going to draw it all the way around. So we have a net charge of positive out, negative in. And by the way, this environment, this polar environment of opposites on one side from the other, is just along the edge of this membrane. It's not way out here in the extracellular fluid. It's just right around just the very edge of that membrane. So the first thing that I'm going to define today is something called a graded potential. So review this in your book. I will define it and describe to you what it is. This is a localized, short-lived change in membrane potential or polarity. We call it graded, and I'm using air quotes when I say graded because the magnitude of that potential or that impulse is directly related to the stimulus strength. So what is here's in reality what a graded potential is and does. So we're looking at this neuron at rest. If another neuron, let's say, here's the end of that neuron's axon terminals, right here, stimulates our big neuron, the one that I first drew. So if this neuron up here gives an action potential, releases its chemical onto the dendrites or the cell body of this resting neuron, the first thing that this resting neuron exhibits is a graded potential. And by definition, what that means is the chemical that's released onto the dendrites and around the cell body, the receiving end of this neuron, causes a change in polarity along this membrane. Okay, that's what a graded potential is. Literally what happens is sodium channels open, sodium gates open along this membrane after this neurotransmitter binds to the membrane. These sodium gates open up, sodium ions go into the cell. That causes a relatively mild depolarization all around this membrane. From now on, when I say the word depolarization, I want you to think, whenever you hear depolarization, think sodium ions going into the cell, and that causes a flipping of charge. It causes depolarization. So when I say depolarizing or depolarization, or you read it, A, think of sodium rushing into the cell or going into the cell. B, think of this polar environment becoming less polar. So positive goes this way, negative tends to go this way. That's what this word means. We left off there yesterday with that definition. Now, a graded potential just means when neurotransmitter from a presynaptic cell releases its chemical onto this resting postsynaptic cell, we get a change in polarity, and it acts like ripples in a pond when you throw a stone into a pond. And it emanates from the site of stimulus. So this neuron up here is releasing its neurotransmitter. It's binding to the surface of this resting neuron, and that causes a ripple of depolarization along this membrane from the site of stimulus away from that site. We call that a graded potential. Graded potentials occur at or around the cell body of a neuron from the point of stimulus. If this graded potential reaches the level of threshold of this resting neuron, if threshold is reached, that means if this graded potential that's rippling around here, if it's strong enough to reach this resting cell's threshold, then something happens right here at the axon hillock. Okay? So picture it this way. If you throw a small pebble in a pond, you get small ripples. Here, we're throwing a pebble in the pond, the point of stimulus. We get small ripples, and then they die out quickly. If I throw a larger stone into the pond, in other words, increase the stimulus strength, we get larger ripples of depolarization that go further around. So bigger ripples in the pond that travel further. If I stimulate this cell strongly enough, the analogy is throw a huge rock into that still pond. Those ripples are bigger and they emanate far enough all the way around this cell body to reach this resting cell's threshold. When that occurs, it's like opening up a dam that we can't close anymore. And when threshold is reached from that graded potential, then at the axon hillock, right here where I've made I'll make it a red ring, right here where I've made this red ring at the axon hillock, what happens is sodium gates open up like a dam. They open up really wide, and sodium ions right here literally gush into the cell. They pour into the cell. That is the beginning, the initial stimulus, once this cell reaches threshold, of an action potential or an electrical stimulus. So once threshold is met, an action potential will fire, and that's from massive depolarization right here at the axon hillock. Sodium gates open. Now we can't turn this off. Once threshold is met, this action potential is called an all or none response. Once we open this huge gate at the axon hillock, we can't shut it off. Sodium ions gush into the cell, and then sodium gates open in a linear fashion all the way down this axon. And so sodium ions then rush into the cell in a linear fashion all the way down the axon. What that does is it causes the flipping of membrane charge from positive out. When we started with positive outside, and negative inside all the way around the cell. Once these sodium dams open up and sodium gushes into the cell, we flip the membrane charge to positive in and negative out all the way down the cell. The relative, the net charge becomes positive in and negative out, and that's from these sodium ions rushing in. That is what an electrical stimulus is. That is an action potential. It's the massive depolarization or flipping of charge all the way down this axon in a linear fashion. Okay, so we have now stimulated this cell, caused a large enough graded potential that at the axon hillock, I'm going to circle it in red so you can see it, at the axon hillock, sodium ions, sodium channels open up and sodium ions rush into the cell. We've turned on an electrical stimulus or action potential. We can't stop it now, and it continues all the way down the axon of the cell. That is the firing of an action potential. That is the nerve cell exhibiting an electrical stimulus. So we've completed actually multiple steps. We've completed stimulus, we've completed graded potential, and we've now opened up and completed depolarization, the beginning of an action potential. I need to just layer over this to clean up my drawing. So I'm just going to quickly draw another neuron, and we're going to continue this story. So we've depolarized this cell right here at the axon hillock. Sodium gates have opened all the way down the axon. Sodium has gushed in. That's the act of depolarization. Now, what happens as this depolarizing jumps from gate to gate in a linear fashion in this direction, away from the cell body? Sodium gates then close upstream. As these gates are opening during depolarization, these first gates in a linear fashion start to close. And next, what happens after depolarization is the act of repolarization. So depolarization is always sodium ions rushing in to the cell. During repolarization, potassium ions rush out. So as these sodium gates close, I'll use yellow, potassium gates then take their turn and they start to open in a linear fashion as I'm drawing, and potassium ions rush out of the cell. This is the act of repolarizing the cell. Okay? And the behavior is the same. It happens in a linear fashion, one gate after another. As potassium ions rush out, so much potassium rushes out. Actually, volumetrically, so much potassium is out here now that it reestablishes the original polarity of positive out and negative in. So repolarization reestablishes original membrane polarity of positive out and negative in. The problem is, I shouldn't say the problem, but the challenge is now we've got all these potassium ions outside of the cell and all these sodium ions inside of the cell. So even though we've reestablished original membrane polarity, the ion concentration is mixed up from the beginning of the story. Okay? I do want to, before I go to the next step, this word, hyperpolarization, pay attention to it in your textbook. That just refers to during repolarization of potassium ions flowing out of the cell, so many of those potassium ions flow out that the original polarity values of positive out and negative in gets overshot. So that's what hyperpolarization just refers to. In some cases, oftentimes, so much potassium during repolarization flows out of the cell that we make this temporarily, we make this environment even a stronger positive and negative than what was the original situation of the cell. That's all that hyperpolarization is. It's an act during repolarization. So back to our story. I'm almost finished. We've gone through depolarization. Actually, we've gone through resting membrane potential, graded potential, threshold being met, depolarization occurring at the axon hillock, repolarization occurring after depolarizing. I defined what hyperpolarization means. Now we've got all of these potassium ions out here. Even though it's positive out, negative in, like the beginning of the story, now we've got potassium out and sodium in. And what reestablishes the original ion concentration is the sodium-potassium pump. So at the very end of this story, it's that ATP-driven pump, the sodium-potassium pump, is what reestablishes, and it takes time to do this, especially compared to the first activities, original ion concentration. I know this is language. I know it's confusing language, especially if you've never heard this before. Repolarization reestablishes the original membrane polarity. The sodium-potassium pump reestablishes the original ion concentration. That's important. So that's the whole story. That's how a nerve cell works. If you understand that story, it's many pages in your textbook. Now you can layer on a little bit more detail, but that's the gist of it. If you understand this part, you're way ahead of the game. So 2 things before I erase this board. I'm going to write absolute refractory period and relative refractory period. These are timed events that occur during this story. Absolute refractory period occurs during depolarization. So as those sodium channels are opening up all the way down this axon during depolarization, if we stimulate this cell again during depolarization, it won't respond to another stimulus no matter how strong it is. That's absolute refractory period. Relative refractory period occurs during repolarization. And that's defined as, so while these potassium gates are opening, the yellow, and potassium is rushing out during repolarization, if we stimulate the cell with a second stimulus again, that's stronger than the first one, it will respond and start to depolarize again. So relative refractory period just means the cell will respond to a second stimulus during repolarization if the second stimulus is stronger than the first. All right, I know you have time. You can rewatch this a million times if you still need to digest that. It's a lot, but I need to move on quickly to classification of neurons. We classify neurons both anatomically, structurally, and functionally, physiologically. So let's do the easy one first. Structural classification is just this. This is what neurons basically look like. Multipolar, I'm going to go bipolar, and unipolar. Based on what they look like. Multipolar neurons have 3 or more processes total, branches. They always have one axon, so that means it has 2 or more dendrites. So multipolar are the things that I've been drawing. Typical motor neuron. That's a multipolar neuron because it has one axon and many dendrites. A bipolar neuron is defined as having 2 total processes, 1 dendrite and 1 axon. It would look like this. Here's the dendritic end. Here's the cell body, nucleus, and here's the axon. That's a bipolar neuron. A unipolar neuron looks like a bipolar neuron, but it isn't. Sort of works like this. It's a bit more confusing. I simplify this, but it has one, it's defined, unipolar is defined as one process attached to the cell body. There it is, this little neck thing. And the dendrites, the dendritic direction and the axon direction both use this one process attached to the cell body to do their thing. So for example, an impulse could come in the dendritic end, go up, be processed, and then the depolarization, the action potential could be sent out this side. So multipolar neurons, these are the most common in the human body, most common type of neuron. They're the major type of neuron in the central nervous system. These are basically all motor neurons and most interneurons in the central nervous system. Bipolar neurons are quite rare. So these are common. Number 2, bipolar are not. They're really quite rare. They're typically found in special sense organs, like eyesight, hearing, taste, smell, those things. So they're special sense. These are special sense organs. We find bipolar neurons. Unipolar are somewhere in between. They're not as common as multipolar. They're not as rare as bipolar. They are mostly sensory neurons that take impulses toward the central nervous system. Those afferent neurons tend to be unipolar. And they're found in, these are typically found in ganglia. This word, ganglia, just means clusters of cell bodies. So we have neural ganglia in our peripheral nervous system, mostly of our sensory neurons carrying impulses toward the central nervous system. All right, that's structural classification. Functional, you pretty much already know. So I'm just going to rip through that really quickly. And it's just 1, sensory afferent. You know those functionally carry impulses toward the central nervous system. 2, motor efferent or efferent. You know that those mostly reside in the peripheral nervous system. They use the peripheral nervous system and they carry impulses away from the central nervous system. And 3, what we call interneurons or association neurons. Same thing. These are predominantly found, they're the bridge between the sense number 1 and 2. They're the bridge between sensory and motor, and they're found in the central nervous system. And they're 99% they're the most common functional type. not of the structural type, of the functional type, are interneurons. That's why our brains are so huge. That's where most of our neural tissue volumetrically is located, in the brain and spinal cord, certainly relative to our nerves that emanate out into the peripheral nervous system. All right, very quickly. This is easy business, so I'm going to conduction, that says conduction velocity. How quickly a nerve cell can transmit an electrical impulse is basically dependent on two things. Dependent on axon diameter and degree of myelination. How myelinated the axon fiber is. Axon diameter, the bigger, the faster. The bigger in diameter, the faster it can conduct electrical impulses. Degree of myelination, more equals faster. So less would equal slower. And now we can classify neurons based on these two criteria. And we just call them group A, group B, and group C fibers. Group A fibers, these are the largest in diameter and the most heavily myelinated. So group A fibers are the fastest by far. They conduct impulses up to about 300 miles per hour. If you think about moving from your spinal cord to your fingers at a speed of 300 miles per hour, it happens really fast. Group B fibers are less large, they're smaller in diameter than group A, and they are lightly myelinated. And they conduct impulses. The easiest way to remember this is tenfold. So they conduct impulses about 30 miles per hour. And finally, group C fibers. These fibers are the smallest in diameter, and they are unmyelinated. So they're going to be the slowest. They're about 2 to 3 miles per hour. Now I'm going to give you an idea of where these are located in the human body. Group A fibers are typically somatic, sensory, and somatic motor fibers. So you stick your finger on a hot stove, you jerk it back in a millisecond. Those are somatic, sensory, and motor fibers. The sensory are taking the impulse towards your CNS, the motor fibers away. Group B and C fibers tend to be, first, autonomic fibers. So those are efferent fibers going away from the central nervous system toward our visceral organs. Things like digestion, respiration, blood pressure, those occur much more slowly. The speed of impulses occur much more slowly. The reason is because mostly these deal with the external environment for survival in a lot of ways. This is obviously the internal environment. Things don't need to occur quite as quickly. Couple definitions. That's supposed to be an N. I'm trying to write so fast that I can't spell. So continuous, I'm trying to write the word continuous conduction versus saltatory conduction. This is referring to conduction speed as well. Continuous conduction occurs in unmyelinated nerve fibers, and it has to occur like this. If the axon of this nerve cell is not myelinated as the cell fires an action potential and depolarizes, it has to occur in a linear fashion, gate after gate after gate after gate after gate, all the way down the axon. Picture yourself taking baby steps heel to toe to get from one point to another. That's slow. Saltatory conduction occurs in myelinated neurons, and it's much faster. And what happens is, in a myelinated fiber, so I'm going to myelinate this thing. I'm wrapping insulation, lipid-based insulation from Schwann cells around this axon. And the action potential, which starts here at the axon hillock, it doesn't have to go gate by gate by gate in baby steps. It can literally take giant steps and jump from node of Ranvier to node of Ranvier. That's much faster, much, much faster. All right, now we need to go to synapses and types of synapses. You know now that the word synapse basically just means the junction between one neuron and a postsynaptic cell. In this nervous system lecture, we're mostly talking about from neuron to neuron. So we've got a presynaptic neuron and a postsynaptic neuron. That just means the synapse is the junction. There are this is just definitions, it's just language. There are axodendritic synapses. That just means that the synapse the junction occurs at the synapse. And there are also axosomatic synapses that occur at the soma or the cell body. It doesn't really matter, the result is the same. Whether or not the presynaptic neuron is releasing its neurotransmitter onto dendrites or right onto the cell body, the result is usually the same. Neurons can have up to 10,000 axon terminals at the end. You know, when I draw the end of this thing, I only draw a few, but in life, there can be up to 10,000 of those things. So you can imagine how many postsynaptic synapses that one cell can have. All right, but the reason I said all that business is because I want to review types of synapses. Okay, and there's basically two classes. I'll do this one first because that's the one we're familiar with. Chemical synapses, by definition, just means a neurotransmitter is released into the synaptic cleft and it binds to the postsynaptic cell. Chemical means neurotransmitter is present. You know that that neurotransmitter is stored in vesicles in the axon terminal. There's no physical connection between the presynaptic cell and the postsynaptic cell. It must use neurotransmitter. There's always a synaptic cleft, that space that the neurotransmitter has to diffuse across, and double star, communication is always unidirectional. That means communication is always from the presynaptic cell in one direction to the postsynaptic cell. That all sounds probably intuitive because we've only talked about chemical synapses so far. But now I'm going to bring up the other class of synapses. Those are electrical synapses, and they behave quite differently. These are made up of gap junctions between cells. So there is a physical connection from cell to cell. I don't have time to go through exactly what a gap junction is. That's Bio 1 business. But basically, let's just say we have a cell, any cell here. We've got another cell here. A gap junction is usually some sort of protein connection, physical connection from cell to cell. These protein channels that I've drawn have a name. They're called connexons. C-O-N-N-E-X-O-N-S. Those connexons connect the cytoplasms of the two cells, and they are electrically coupled. That means the flow of ions can move from this membrane or this cell to this one, and vice versa. So there is a physical connection, and that's through those protein junctions called connexons. Electrical flow can occur from cell to cell, and the flow can be bidirectional, can go from either cell to either cell. It's not unidirectional. These have very, very fast transmission because there's a physical connection, typically. They are found largely in the brain and allow for coordinated synchronization. Found in the brain for a very coordinated, meaning going in both directions from cell to cell, a real synchronous type of transmission or communication. Now I need to talk about, I'm going to go back to this and just briefly talk about how chemical neurotransmitter is released from a chemical synapse, the presynaptic cell. So I'm sort of skipping, may not be going in direct order of your textbook, but that's okay because I need to hit on all of this stuff. You know a little bit about neurotransmitter release, but not the whole story. So I'm going to draw a really big axon terminal. And I'm going to go through this in steps. Step 1, action potential travels down this membrane, aka depolarization. So it arrives at the axon terminal, the end here. Voltage-gated calcium channels open up. So this electrical activity here, this depolarization, causes voltage-gated, because it's an electrical activity that's causing it to open. A voltage-gated channel, calcium channel, opens up, and calcium rushes into the cell. Calcium ions from it, the extracellular space, rush in. We know that neurotransmitter is stored in these big bubbles called vesicles. So there's a bunch of, the specks are the chemical, the neurotransmitter, and this bubble is a vesicle. When calcium enters the cell from this electrically gated channel, from this action potential coming down, calcium binds to a protein on the surface of this vesicle. That protein is called a synaptotagmin. I'll write it out, don't worry. So there's the protein on the surface. This long word, synaptotagmin, is just a protein on the surface of the vesicle that has a binding site for calcium ions. Then, after calcium binds to this synaptotagmin, it interacts with what's called a snare protein. So out here is a snare protein, and it's all capitals because it's an abbreviation. Every time, whenever something binds to something else in the human body, almost always it causes a change of shape. So when calcium binds to this synaptotagmin, this whole thing changes shape and it moves this vesicle. Now this can bind to a snare protein and it changes shape again, and it literally causes this whole vesicle then to migrate to the edge of the axon terminal. It still has its proteins on it, and they're bound to the snare proteins. I'm not going to ask you what snare stands for. It's a synaptotagmin. The snare, I think, is N. It stands for the chemicals that it's made of and enzymes. I forget myself offhand what it stands for. I'll think of it before this is over. Nevertheless, calcium binds to the synaptotagmin. That then complex binds to a snare protein. Then it migrates to the edge of the terminal of the axon terminal, where it binds to the fusion of the plasma membrane occurs, and neurotransmitter is released into this space. So we've got step 1, step 2, step 3, step 4, it migrates and releases into the synaptic cleft. That's how the whole business really works in detail. This is the rate limiting step. That means this, when I was a student and my instructor asked me, what do you think of neurotransmitter being released, migrating across the synaptic cleft and binding to receptors on this postsynaptic cell? What step takes the longest? And almost everybody in the class, including myself, raised their hand when he said diffusing across the synaptic cleft. That is not what takes the longest. This process here takes the longest time. For its neurotransmitter release takes longer than the diffusion process across the cell. That is called synaptic delay. So when you read that term, synaptic delay, that refers to this process that I just described in here, and it's the longest step time-wise in transmission of an electrical impulse. All right, I'm looking at my clock. Hopefully I'll get through most of this business. We need to go on to postsynaptic potentials and synaptic integration. What this means is what's happening at the cell that's receiving the electrical stimulus. Good question. No, there's a whole, I just got a question about, is neurotransmitter always acetylcholine? I'll talk about that later after you take this next quiz. We're going to continue with the nervous system, and there's all different classes of, there's like 20 some to 50 different classes of neurotransmitters. And we haven't discovered all of them in the human body. So there's many more neurotransmitters than you could possibly learn. And I don't know all of them off the top of my head. I know many of them. And they're based on typically their chemical classes. And then they're also grouped on how they act. We're going to stick with acetylcholine now. I'll talk about different types later after the next quiz. So postsynaptic potentials refers to what is happening at the cell that receives that neurotransmitter, okay? So there are two different types, basically. There's what we call EPSPs. That stands for excitatory postsynaptic potentials. That means when the neurotransmitter binds to the receiving cell, it causes an excitatory response. So this stands for excitatory postsynaptic potential. And here's really what happens. Step 1, the neurotransmitter, when it binds, it causes, in EPSPs, excitatory responses, it causes depolarization. So it depolarizes the membrane of the receiving cell. So always equate depolarizing with excitatory, okay? Step one. And you know the rest of most of this story. Sodium, potassium diffuse in and out of the cell. That's from a chemically gated ion channel. A net depolarization occurs, first a graded potential, and then if threshold is reached, then an action potential being generated in the postsynaptic cell. The function of an excitatory postsynaptic potential in general is this. The main function is to help, that's supposed to be an H, is to help trigger an action potential. I'm going to put, obviously, at postsynaptic cell. So there's a really Reader's Digest version on what an EPSP is, an excitatory postsynaptic potential. That's what we've been talking about using as our examples all this time. It's excitatory, it causes depolarization, which by definition makes it excitatory. And the main goal is to help trigger an action potential at the axon hillock of the postsynaptic cell. Now that's in contrast to an IPSP, which is inhibitory. So sometimes the release of a neurotransmitter onto a postsynaptic cell, onto another cell, is not excitatory, it can be inhibitory. And here's the Reader's Digest version of this. They inhibit activity at the postsynaptic cell, so it inhibits the ability of the postsynaptic cell to depolarize and fire an action potential. So the opposite occurs. Now we have to really dissect very quickly in the next minute or so what that means. Most inhibitory postsynaptic potentials do this by not depolarizing the membrane of the receiving cell, by hyperpolarizing the postsynaptic membrane. So what they're doing is they're taking the cell, the receiving cell, away from, further away from threshold, not closer to threshold. So remember, if we have a membrane here, sodium out, potassium in, it's polarized, positive out, negative in. An inhibitory postsynaptic potential, when it releases its neurotransmitter onto this cell, would make this more negative and this more positive, taking it away from depolarizing. Remember, depolarizing means positive, negative. We get closer to flipping this charge. Inhibitory, always think hyperpolarizing and taking this receiving cell further away from threshold. So all of this business, what does it tell us? At the end, the summation of this is what can be concluded from this is the action of a neurotransmitter on any postsynaptic cell is not dependent on the neurotransmitter itself. Is not dependent on the type of neurotransmitter or the chemical makeup of the neurotransmitter. It is dependent on the type of receptor at the postsynaptic cell. Classic example is this. The neurotransmitter, the only one that we've been talking about, acetylcholine. You know, when acetylcholine is released onto skeletal muscle from our last unit, it's excitatory. It causes that muscle cell to contract. The same neurotransmitter, when it's released, the exact same neurotransmitter, acetylcholine, when it's released onto cardiac muscle, guess what? It's inhibitory. It takes those cells further away from threshold, and therefore further away from contracting, and that actually slows heart rate. It doesn't speed it up. That tells us that the action of a neurotransmitter is not dependent on this, it's dependent on the type of receptors on the postsynaptic cell. All right, some quick definitions before I call this a day. I'm not going to get to the whole thing. I do want you to review this in your book. I'm just writing summation. That's supposed to be SUMMATION, summation of neurons. We briefly hit on this when we were talking about graded muscle responses and what can cause them. But there is a temporal summation, just like we listed in the last unit. That means increasing the rate of neurotransmitter release. And there is also spatial summation. When we talked about it in a muscle cell, we talked about just innervating more than one muscle fiber. In this case, this is talking about increasing the rate of neurotransmitter release, firing that neuron more quickly over a span of time. This is referring to the number of neuron stimulation. So this is rate of release, and this one is the number of neurons. If we have, for example, if we just have two axon terminals stimulating the postsynaptic cell, contrasting with if we had, let's say, 25 axon terminals stimulating the same postsynaptic cell, that's going to cause a summation response on this postsynaptic cell, whatever it is, and it will reach its threshold more quickly. So in this one, we increase the rate of release of neurotransmitter. This one, we increase the number of neurons stimulating the postsynaptic cell. Both of those may increase, depending on the situation, or decrease the stimulus, the ability of that cell, postsynaptic cell, to reach threshold. Okay, I already talked about neurotransmitter effects. I'm going to just briefly mention neurotransmitter receptors. So the types of receptors at the postsynaptic cell. There are things called channel-linked receptors. I'm just going to tell you about these. You can read about them in your book. These are what your book calls ligand-gated channels. That word, just think protein, a protein gate on a membrane that allows things, that it opens and closes, it allows things to move through. So when you see the word, it's a signaling chemical that binds to membrane receptors and then causes it to change shape and open up or close. So there are channel-linked receptors. The protein receptor changes shape when the ligand actually binds to it. So the ligand is sort of like it's a signaling chemical is what it is, and it binds to a protein receptor on the membrane. The whole thing changes shape, and that gate opens or closes. That's what we call a channel-linked receptor. This type always receives neurotransmitter. It can only always receive neurotransmitter. It can be excitatory, opens or closes, so it can be excitatory or inhibitory. I'm getting ahead of myself with my spelling because I'm in a hurry. Now let's talk about the second major class, which are G protein linked. We'll examine these more at the very end of this course when we talk about the endocrine system. These are a bit more complicated in some ways. I'm just going to tell you the main differences. So these receptors they're much more indirect, and they're also much slower than channel linked. So they're also called, they have another name, and I'll tell you why. Metabotropic, it's all one word, receptors, because they change the metabolic behavior of the receiving cell. That's why. These are used in some learning processes in the central nervous system. So when you learn how to do a new task, and it's a slow process, so they're slower. We use them in what we call learning processes in the central nervous system. Basically, I'm going to draw out here what a G protein linked receptor means is on the surface of this cell, there's a receptor that binds the neurotransmitter, but then it's linked to a protein on the inside called a G protein. These G proteins then are typically linked, physically linked to what we call a second messenger. So they're also called second messenger systems. I'm going to go over this in the endocrine system. The second messenger can be something like cyclic AMPs, cyclic GMP, calcium ions. There's all different types of second messengers. What they usually do is they activate kinases inside of the cell, which regulate, speed up metabolism or slow it down usually. So review these in a little bit more detail, but that's the gist of what they are. And those are the major points of them. Can't believe I did it, but I got through, I think, everything on my list. I know this was a lot. I'm going to cut off my recording, and then I will let you ask any questions you want.